Cas9 recruiting activator domains, the RNA is recruiting the activator domain by interacting with a second protein that's expressed. And so Feng appended in his CRISPR-A system VP64 domain directly to dCas9, and then he also appended 2 MS2 stem loops internally on the sgRNA in a manner that was informed by the crystal structure. So this was like really nice piece of work. They waited until they had the crystal structure and then didn't just blindly stick these MS2 stems onto the sgRNA sort of a unguided fashion like we did, but instead said we're going to use the structure to figure out where we can append these and not impact the function of the sgRNA. And stuck to there, then showed that this doesn't compromise the activity of sgRNA loading into Cas9 or dCas9. If you co-express an MCP coat protein that binds to these MS2 stem loops, fused to 2 activation domains called a P65 domain. This is derived from the endogenous NF-kappa B pathway in human cells, along with the HSF1 activation domain, which is also an endogenous activation domain. You're now with a single sgRNA recruiting 3 different activation domains. So this one's recruited with the protein, and these 2 are recruited by the RNA. And he showed in this paper that this works quite nicely to activate transcription of endogenous genes, and it is another way to multiplex your ability to deliver different domains using a dCas9 and a single sgRNA. We had a similar idea at UCSF. We didn't have the crystal structure at the time, so we just appended these stem loops to the C-terminus of an sgRNA. This is work from Jesse Zaletan, who has his own lab at University of Washington now, but the work was done in Wendell Lim's lab at UCSF. And what he was able to show is that, as I've shown you previously, we can use dCas9 to control transcription, either just by using dCas9 alone or by appending protein domains to dCas9. And in this setting, the guide RNA that you express only encodes the locus that you're targeting. It doesn't do anything functionally. In contrast, if you start to append these RNA domains to an sgRNA, now the guide RNA encodes both the locus that you want to target and the action that you want to perform. So you can append different hairpin structures to an sgRNA that are targeted to different genomic locations, and with one dCas9 will activate and repress transcription at different loci. So that's shown here, where you have this purple stem loop that recruits this purple protein fused to this green effector domain, and that's controlled by this gray dCas9. So this can all be targeted to gene X. And then in the same cell with the same dCas9, you don't need an orthogonal dCas9, you can append a different sgRNA with a different stem loop that recruits a different protein that's fused to a different effector domain to do something different at a different locus. So it just increases your ability to use one CRISPR system to control the expression of multiple genes in different ways, in a way that you can imagine could be really useful for a lot of applications. And so the question was, does this work? Jesse fused 3 different stem loops to the sgRNA and showed that each of these, when you co-express a protein that recognizes a stem loop fused to an effector domain, can activate transcription of a reporter gene in yeast. So they're quantitatively a bit distinct, but you can see that if you co-express all these components together, each of these will activate expression of this Venus reporter. This can be used he showed that there was no crosstalk between binding pairs, so you can co-express these in the same cell, and they don't encode the wrong functions at the wrong loci. So another way of saying that is they're completely orthogonal. You can use this same sgRNA scaffold general structure to recruit, to amplify your signal. So you can append multiple stem loops to one sgRNA to either recruit 1, 2, or 3 of these effector proteins. And that can have an additive effect on transcription. And then you can also append 2 different RNA stem loops to the same sgRNA. So here we're appending an MS2 stem loop and a PP7 to the same sgRNA, and in this manner you can recruit either 2 of the same domain using an RNA, or 2 different domains using one RNA to control transcription. This also works in human cells, so we used the MS2 system, or this COM-CRAB system, to show that in human cells with one DCAS9, we can control the expression of two genes and turn one off and turn the other on. So that's shown here, where we're activating expression of CXCR4, or we're turning off expression of B4-GALAT-T1, or we can turn on one gene and turn off the other gene at the same time using one DCAS9 scheme. So it just increases your flexibility for controlling transcription across the genome. This works for metabolic control, which you could imagine, like the ability to control multiple genes dynamically could be really useful for metabolic imaging. And so in yeast, Jesse was able to show that he could tune the output of this violacin pathway. This is a fairly complex pathway that has it's a 5-gene pathway, so violacin A through D, and depending on which gene is activated, you get production of these different metabolites that have different colors. So this one's pinkish, that one's purple, yellowish, and sort of a cyan. And so what Jesse was interested in is whether you could use a single DCAS9 to turn off and on different steps in the pathway and tune bioproduction of these different metabolites. So this works, and he showed that in the absence of any sgRNAs, you get this the pathway is off, more or less. If you start to activate the pathway, you produce this PV intermediate. If you activate the first two steps, you produce V. If you activate this first step and turn off D, you now produce PDV, which is shown on these HPLC curves as different peaks for these metabolic intermediates. And then if you activate the first two steps and turn off the last step, rather than producing this, you produce that. And so sort of a toy experiment, but nice ability to show that we can control expression of more than one gene to specify the output of a pathway in a organism that is thought of widely as a fairly useful metabolic, organism for bioproduction. So I think some of the questions that remained at this point, and some of these will be answered by Mike, are can we manipulate pathways in the same way that I just showed you for yeast in human cells? Can we use CRISPR-RNA to control expression of all of the genes encoded in the genome and in that way build a functional genomics platform? I sort of already showed you this, which is can we use CRISPR-RNA to control the expression of non-coding RNAs and try to figure out the expression of the many non-coding RNAs that are annotated in mammalian genomes. It's hard to study a non-coding RNA with Cas9 because non-coding RNAs don't have an open reading frame, so there's nothing really to disrupt in the same way that most people are doing genome editing. If you want to disrupt a non-coding RNA with Cas9, you have to delete a large region of the genome. That's both less efficient and also can have unintended consequences if you delete additional regulatory regions. So we think that CRISPR is like a very surgical way to just turn off transcription without disrupting large chunks of the genome, and is useful for studying non-coding RNAs. And then we'd like to start to move towards higher order perturbations where we're controlling the expression of sets of genes. So I'll end this section by just saying work at UCSF. We've done a lot of genome scale screens, and this is sort of impinging on Mike, but it's for one specific purpose. Essentially, we've done, this is an outdated slide, we've done tons of screens. This gives us a lot of data which we can use to train more and more sophisticated machine learning algorithms. So this is depicted in a schematic here, what we've done, but essentially the output is a more and more sophisticated algorithm that predicts which sgRNAs are going to be highly active for controlling transcription in mammalian cells. And so this is published. I put this slide up only as a resource to say if you're trying to do anything like this, the algorithm that predicts sgRNAs is really important, and we're a little biased, but we think that our current algorithm described by Max Horlbeck in this eLife paper is the best one out there. You can use this algorithm, the sgRNA predictions, with any of the CRISPR-A systems that are described by any lab. So it's not restrictive to our CRISPR-A system. You can use it with the Broads or with George Church's. But the protospacer predictions are very, very useful in our hands. And just as an illustration of this, when we tested this algorithm on non-coding RNAs, we can see that by qPCR We're targeting non-coding RNAs that have no phenotype, so we're just purely testing our algorithm here. The vast majority are highly active, and most of them give you robust repression of your target gene. Yeah.
They study the location of transcription starts, so it's not just predicting the sequence, but the location.
Absolutely. The location is really, really critical. So there are features of the machine learning algorithm that are general, and that would be useful for predicting active guides for Cas9 or dCas9, and they relate largely to transcription of the guides in human cells and folding and loading into dCas9 or Cas9, so those would be applicable for any experiment using CRISPR. But then there are also a lot of features to this algorithm that are specific for either CRISPR-I or CRISPR-A, where we're controlling transcription. Those largely relate to, as Dana pointed out, where you want to target these complexes relative to the transcription start sites of genes. So our systems are fairly short-range activators and repressors, and so you need an accurate annotation of the transcription start site of the gene to be able to turn it on or off. Another feature that goes into this algorithm, I'm incredibly proud of Max. He's awesome. This is actually Horlbeck eLive 2016B. There's an A that is really useful. It describes his finding that both Cas9 and dCas9 don't seem to bind either in vitro or in vivo at sites that have a nucleosome occupied. Sorry, that's a very backwards way of saying that. Cas9 can't bind DNA if there's a nucleosome present. So until 2013, Cas9 had never seen a eukaryotic genome. It had never seen DNA wrapped around a eukaryotic canonical nucleosome. And what Max, together with collaborators from Berkeley, showed both in vitro and in vivo is that if you have DNA wrapped around a nucleosome, Cas9 just doesn't bind at all. So if it doesn't bind, it can't cut, can't control transcription, it can't do anything. Fortunately, within the human genome, there's a lot of nucleosome breathing or remodeling, and so the average site across the genome at some frequency can be bound by Cas9 and dCas9. But for controlling transcription, in all eukaryotes, the nucleosome positioning around the transcription start site is fairly well phased. So if you look at a well-annotated TS transcription start site, you can see phased information for nucleosomes downstream from the transcription start site. And so if we just avoid those phased nucleosome positions, we increase our enrichment for active guides that control transcription. Unfortunately for Cas9, nucleosomes aren't phased away from a transcription start site, so the coding exons for most genes are too far from a transcription start site to give us information on avoiding nucleosome positioning, but it is still something to consider for other more directed experiments. Okay, so everything I've talked about so far today is about using dCas9 to turn transcription on or off using a variety of domains. So we think about this as sort of controlling the transcriptome. We are editing the epigenome, but we're not doing it enzymatically, and we're not. Our focus up until this point had been more on the ability to control transcription for reasons I don't entirely have time to get into. But a lot of other labs have been interested in using dCas9 as an easy-to-use RNA-guided DNA binding platform to surgically control epigenetic marks. And so there's been a lot of really cool work from a number of labs over the last 2 years that I want to just summarize over the next 15 or so minutes. So I showed a slide like this yesterday. Basically, my only point here is that the eukaryotic nucleus is really complicated. You have DNA wrapped around histones. The histones have a histone tail, and these tails are highly modified with a variety of methylation marks and acetylation marks, as well as more exotic derivatives like crotonylation and other things, phosphorylation. But the overall integration of all of these marks at each nucleosome dictates transcription of much of the human genome, or much of any mammalian genome. And so this is a really active field of research. You see all kinds of papers saying, this mark at this site does this, whatever. But unfortunately, for the most part, the field of epigenetics up until now has been stuck with either using toy loci, where we have investigated, for years or decades one locus and how the chromatin marks at that locus dictate transcription, or we've used, sort of retrospective techniques like ChIP-seq, where you fix a cell, you pull things out, and you have to correlate the presence of different marks with different transcription statuses in human cells. And so what we'd really like to be able to do is, in a living human cell, edit a mark at a specific site and ask how that turns transcription on or off dynamically over time, like through differentiation or through some drug treatment. And so labs have started to do this. So building off of sort of our initial work in yeast and human cells, people have used dCas9 and appended on additional domains. And so some of this work is really all from 2016, where people have, a couple different groups have fused on DNA demethylases to remove methyl marks off of DNA. So I talked entirely about histone marks, but DNA also has epigenetic changes, just methylation, sorry, not changes, that can be dynamically deposited or removed to control transcription. And so to remove or deposit DNA, people have created dCas9 demethylase fusions and dCas9 methyltransferase fusions. People have also fused on histone acetyltransferases and histone demethylases to edit the marks on histones. And so I'll just talk you through some of this very briefly. One of the first papers was from Renee Mayer's lab at UMass, published in 2015, where they fused, actually, this is Charles Gershbach's lab, not Renee Mayer, so sorry, backwards. They fused the P300 histone acetyltransferase domain to dCas9 and asked whether they could deposit acetylation marks at an endogenous gene using by targeting dCas9 fused to P300 domain to that locus. And so they showed this, that they could deposit acetylation marks by ChIP-seq, but also just asked whether deposition of increased acetylation would activate transcription. So that's shown here, and you can see that they can quite robustly use these dCas9 P300 domains to deposit acetylation at these three endogenous genes, and that turns on gene expression. So rather than, like we had shown previously, use scaffolds, now they're directly editing the epigenome to activate transcription. One of the coolest things I think about moving beyond scaffolds towards actual epigenetic enzymes is that we can control the chromatin or DNA epigenetic marks.